All right, so we need to talk about anaerobic cellular respiration. Now that we've talked about aerobic, remember that with aerobic, oxygen was used. Remember that the O2 would come and pick up carbons, and that was what helped to break down the glucose. Well, anaerobic cellular respiration is a process that happens in cells when there's no oxygen available. So that can mean in plant cells when there's no oxygen available, in animal cells when there's no oxygen available, fungus, bacteria, any type of cell where at that moment there is no oxygen available. What's going to happen is that after the cell has gone through glycolysis, instead of the 2-pyruvate moving into the mitochondrion, it's going to sense that there's no oxygen available. And so what's going to happen is that right there in the cytoplasm, alcoholic fermentation or lactic acid fermentation are going to happen. So alcoholic fermentation occurs in plant cells. Ooh, my pen went away. Let me get my pen back. Okay. All right, so alcoholic fermentation occurs in plant cells, in protists, and in most bacteria. Okay, so these types of cells, when there's no oxygen available for them to go into aerobic cellular respiration, are instead going to go through alcoholic fermentation. Okay, we call it alcoholic fermentation because an alcohol molecule is one of the molecules made at the end. Okay, so it starts off just like aerobic cellular respiration. So glycolysis is going to happen. Notice that there's no oxygen required for glycolysis, right? Okay, so just to walk back through glycolysis, we have a glucose molecule, which remember, a glucose molecule doesn't look like this. It's in a ring and it's got oxygens and hydrogens attached, but just for picture simplicity's sake, we made it just a strand of carbons, and we're just imagining that all the oxygens and hydrogens are there. But I did have to draw 2 of the hydrogens that are on it because NAD has to come and pick those up. So 2 NADs come and they pick up 2 of the hydrogens. That leaves an open binding site for phosphates to bond. So ATP is going to come and drop off 2 phosphates. Okay, now we have 2 ADP molecules, and we've got a glucose molecule with 2 phosphates attached. Those phosphates binding makes glucose very unstable, so it's going to break apart in half. And so now we have like a half of glucose with a phosphate attached here and a half of glucose with a phosphate attached here. Phosphate has done its job. So ADP is going to come and pick back up its phosphate. So we got our 2 ATPs back. Now we have 2 pyruvates, which are just 2 basically halves of glucose that are missing a couple of hydrogens. So this is basically like half of glucose missing a hydrogen, half of glucose missing a hydrogen here. Okay. Now remember that when right after we get pyruvate, that 2 more ATP are going to be made. And we didn't learn exactly what happens here, but we know that during glycolysis, the last thing that happens is that 2 ATP are made. So since we used 2 here, we made 2 here, and we made 2 more here, we get 2 net ATP through glycolysis. So this is, glycolysis happens to every single glucose molecule no matter what. Okay, as long as there's ATP available, this process will happen. Now, once we get to this point right here, okay, so once we get to the end of glycolysis, if there is oxygen present, then we know that this pyruvate is going to move into the mitochondrion. If there is no oxygen present, then instead, another process is going to happen right here in the cytoplasm. So basically, pyruvate is not a molecule that the cell can store, and so it can't just make pyruvate and then just let it sit there. Okay, so if the pyruvate can't move into the mitochondrion to go through aerobic cellular respiration because there's no oxygen available, then instead the pyruvate has to be turned into a molecule that can be stored. So what happens is that if no oxygen is available and glycolysis has occurred in plant cells or protists or most bacteria, then what happens is this last step is really the alcoholic fermentation part. So what happens is that these 2 NADHs that were made earlier that would normally go to the electron transport chain, instead of going to the electron transport chain, they come back around and give their hydrogens back. Okay, so they give their hydrogens back to the pyruvates. Okay, so which now we have C6H12O6 because we still have all our carbons that we had to start with. We still have 6 between the two pyruvates. We still have all the oxygens. We didn't take any oxygens off. All that was taken off was 2 hydrogens. So NADH brings back the hydrogen. So now we have C6H12O6 between these two pyruvate molecules. We still have all the makings of a glucose molecule. But what happens is that C6H12O6 actually arranges into 2 carbon dioxide molecules and 2 ethanol molecules. Okay, so remember, we've got all the makings of glucose right here because all we did was break it apart. We still have all the parts. So C6H12O6 is what we started out with up here. Okay, when we get to the end, because we got our hydrogens back, we still have all the makings of that. We've just got to rearrange it into a molecule that can be stored. So what's going to happen here is that we're going to make 2 CO2s and 2 ethanol molecules. As you can see, that means that if we have 2 CO2s, then we multiply the 2 times the C. So for carbon, we've got 2 here, and then 2 times 2. So we come out with 6 carbons, exactly what we needed. Okay. And we've got 2 CO2 molecules, so that means we have 2 sets of O2 molecules, so 2 times 2. We got 4 oxygen molecules from carbon dioxide, and we've got 2 times 0, 2 times oxygen is just 2. Okay, and we come out with 6, which is exactly what we wanted. And then we have hydrogen, which we haven't done yet. And we got our 2 hydrogens back, which means that we have all the hydrogens that we need. So 2 times 6 hydrogens is going to give us 12 hydrogens. Okay, so this is basically the whole point of this is that we do get a couple of ATP molecules out of it. Okay, now we didn't make anywhere near as much ATP as we would have if we could do aerobic, but there was just no oxygen present. And so the best thing that we can do is make another molecule that can be stored out of this. Okay, so that's what happened. This is what happens in plant cells, protist cells, bacteria cells if there's no oxygen present once we get through with glycolysis. Okay, we basically just make these molecules out of what we're left with. Now, if there's no oxygen available in animal cells, then what's going to happen is a process called lactic acid fermentation. You probably heard of lactic acid when you have heard about like muscle soreness and things like that. There are studies that are trying to relate lactic acid to muscle soreness, a buildup in your muscles of that. So it's possible that that's related. But again, glycolysis occurs because that always happens. So glucose is going to have 2 of its hydrogens taken away. So now we got a glucose that's ready for 2 phosphates to bond. And then those phosphates are going to make that molecule very unstable. The molecule breaks down the middle. Now we have a half a glucose with a phosphate, half a glucose with a phosphate. Phosphate's done its job, so ADPs are going to come and pick its phosphates back up. So we used 2 ATP here. We made 2 ATP here. We're at net 0. Okay, we've got 2 pyruvate molecules. Remember that once we have these pyruvate molecules, 2 more ATP are going to be made. It's a process we didn't learn, but that's okay. Just know that 2 more ATP are made at the very end of glycolysis. So this is the end of glycolysis right after those ATP are made. So we are at a net of 2 ATP. Glycolysis. Okay, now we finished glycolysis. There's no oxygen available, which means that these pyruvates can't move into the mitochondrion to go through aerobic cellular respiration. So instead, we're going to make the pyruvates into a storage molecule. So we're going to take each pyruvate and we're going to put the hydrogen that we took off up here back onto each of the pyruvates. So now we have a pyruvate with a hydrogen, pyruvate with a hydrogen. Okay, pyruvate with a hydrogen attached is called lactic acid. And this is the molecule that we can store for later. Lactic acid, it's not the most efficient thing that our bodies can do. With lactic acid, it can be stored in our muscles only for a certain amount of time. Eventually, there's a process that has to happen to take the lactic acid and convert it back into glucose. But that's a process that requires ATP, requires oxygen. And so this is definitely not the most effective method of making ATP. and making energy to fuel our systems in our body, definitely the more effective method is aerobic, to make more ATPs. But this is what happens when our body needs really quick bursts of energy, because glycolysis can happen really, really, really quickly and make 2 ATPs every time. Okay, so again, if aerobic cellular respiration is not occurring, if there's no oxygen available, then this is what happens in animal cells if that occurs, and this is what happens in plant cells if that occurs. Okay, so make sure that you watched the aerobic cellular respiration video before this. Make sure you're taking notes. Again, you could have printed this sheet and taken extra notes on that. Any of that's fine. Make sure you bring it to class on Monday.